Hey everybody, in today's video, we're going to talk about an introduction to proteins and amino acids. We're going to talk about protein structure, functions, amino acids, how do they join up together to form proteins, and what are the different kinds of amino acids that we have, and what are their properties, okay? If you enjoy the video, if you truly like it, if it helps you, please consider subscribing, consider liking, and let's get into it. So, when it comes to protein function, okay, they function in many different avenues when it comes to the cell. They can have enzymatic functions, they can be storage proteins, they can be hormonal proteins, okay? So one example, for example, is like thyroid hormone. It's made up of a whole bunch of tyrosines, okay? Motor proteins as well, like basically 80% of your skeletal myocyte is just proteins, okay? Like these sarcomeres, the actin, the myosin, they're all proteins. It can be defense proteins like immunoglobulins, okay? Or, your IgA antibodies, IgG, all that stuff. They're all proteins. They can be transport proteins, okay? So ion channels. You guys have heard of the sodium potassium pumps. That's also protein. Receptor proteins as well. Okay, so for example, your acetylcholine receptors on your skeletal muscles, they are also proteins. But not only that, they can also function as structural proteins, like collagen in your connective tissue, which is found pretty much everywhere in like every tissue. So you can see the vast function of proteins, and that's why it's essential to talk about them when it comes to nutrition. Now, another thing is, how do you actually make a protein? Well, you guys know that inside of your cell you have the DNA in the nucleus, and through various mechanisms you can go into the nucleus, take this DNA, and transcribe it into something called an RNA. This RNA will then go and interact with some ribosomes on the endoplasmic reticulum or free ribosomes and get translated into a protein. Here is a protein. But what is a protein actually made out of? Well, we all know that a protein has this carboxyl end, so a COOH end, and it has this amino end, okay? But where are these coming from? And what are the kind of like, beads that are just on this string right now, the string of protein? Well, I'll tell you what they are. If we take a little zoom inside, okay, we will notice that all these guys are is just an amino acid sequence. An amino acid is the most basic structure of a protein, okay? It's literally just these little molecules. For example, tyrosine is an amino acid, serine is another one, glycine is another one, okay? And they're just strung together. They're literally just strung together. Now, what do they look like? What would they look like? So they look like this, right over here. Here's the most basic structure of an amino acid, the generic structure. There's an alpha carbon in the middle, okay? And there is a COH, or a carboxyl end, okay? So this is a carboxy group, or carboxy end. And there's an amino end, okay? With a nitrogen bounded to two hydrogens here, okay? And of course, like I said, the alpha carbon. But stemming from that alpha carbon, there is something called an R group. An R group is a variable group. It's a side chain, if you will. And this right here is the only group that is going to be differing between all the different types of amino acids that we're going to be talking about. This R group not only is different, but because of the fact that it's different between the amino acids, it can determine the structure, the function that the amino acid will have, and in turn, what would happen if we put all this amino, and again, the function that protein will have once we incorporate different kinds of amino acids with different kinds of side chains in them. Okay, so that's what the basic structure of an amino acid look like, okay? And don't worry, we'll get into the structure of each amino acid later, but it's important to understand the basic structure first, where we have a carboxy, an amino end, and the R side chain that is variable between all the different amino acids, okay? Now, let's come back to this, okay? So over here we have an amino acid, an amino acid, an amino acid. They're just kind of strung together along this bead. How are they connected to one another? Well, they're connected to one another with this peptide bond. Well, what does a peptide bond look like? Well, let's take an example. Let's take this amino acid glycine and another amino acid called alanine. When you take this glycine molecule, right, here's that basic structure, and the alpha carbon, all right, the carboxyl, and the amino group, okay? And then some variable chain right there, okay? But the point is, this peptide bond is formed when the hydroxyl group on the carboxyl end of one amino acid comes together with the hydrogen of the amino end of another amino acid. So to even simplify it more, it basically occurs when the carboxy end, with the hydroxyl on the carboxy end and the hydrogen on the amino end of another amino acid come together, right? You look at these two, now smush them together. If you smush these two together, what do you get? You get a water molecule, all right? And after you get that water molecule, that's out of the reaction now, and these two amino acids are now joined to one another. They're now joined to one another right over here, okay? That's right there, this little bond right there, that's a peptide bond, okay? That's a peptide bond, and that's what this is. That's a peptide bond right there. And out of that reaction, we get a water molecule, all right? Now, this reaction is called a condensation reaction. Why? Because we're generating water. The other way, if you go the other way and you want to actually break this down, break this peptide down, you get a hydrolysis reaction. Why? Because I'm adding water into that reaction so I can lyse it. I can cut this dipeptide that I created and create the individual amino acid molecules. Okay? Alrighty. And again, when we create this peptide, this is technically called a dipeptide. Of course, there's other ones like tripeptides, there's oligopeptides, and there's polypeptides. And for your knowledge here, dipeptide is 2, tripeptide is 3, oligopeptide is less than 10 amino acids long, and a polypeptide is greater than 10 amino acids. Okay? Okay, now you guys know how the protein is made inside of the body, all right? What is it actually made out of, and how are these guys actually being held together in the most basic structure? Another thing I'll tell you is that the structure of the amino acid can kind of change, not really, but it might slightly change based on the pH that it is in. So you guys know that a low pH environment means that we have a lot of these protons, okay, these hydrogen ions. And a high pH environment means that we have very little of them, okay? So what happens to the amino acid in a low pH? Well, if this amino acid is put inside of a low pH, all right, where there's a lot of protons just kind of circulating around it, it will act as a buffer. It'll act as a buffer, and it will actually take up hydrogen. So look at it. Beforehand, it looked like this, right? NH2, COOH, okay? But now look at it. It might take up some protons to the R group. It might take up some protons to the NH2, making it an NH3. It might take up some protons, turning a COO into a COOH. Okay? What happens when I put it in a high pH environment? Well, in a high pH environment, this is what it would look like. It will actually start donating protons. Okay? So right here, this amino group, it'll donate the hydrogen that it had here. This guy, it'll donate the hydrogen that it had here. Okay? And the R group, it'll donate the hydrogen that it may or may not have had to this environment. Okay? So you can see how this amino acid can change to act like a buffer or not, depending on the pH that it is in, okay? And this tells you something. This means that proteins, okay, can act as buffers, okay? Buffers for what? Buffers for the pH, okay? That's great. All right, now a little more about the protein structure. So we know how we make the protein now, and we know that it's just a string of amino acids, okay? And we know that this string of amino acids is held together by a peptide bond. But you know that's not all. When the protein is made, it will assume a very, very specific shape, and that extremely specific shape will conform to its function, okay? Remember, structure determines function. Now, what is the first shape that it will take? Well, the first thing is called a primary structure, or the first structure it'll take, and it's literally just those amino acids that we talked about strung together in with the help of peptide bonds, okay? Now this one, for example, is a polypeptide chain, which I told you is 10 plus amino acids long. And you can see, again, it's just made up of those peptide bonds. But a protein can take a secondary structure as well. And a secondary structure involves two things. It can take an alpha helical structure, okay, where it kind of is like this coiled shape, all right? Or it can take a beta pleated structure, where the protein is kind of like stretched out, all right? What kind of determines this? Well, this is just the based on the hydrogen bonding, mainly. There are other things, but mainly this is due to hydrogen bonding of the actual amino acid backbones here, okay? Being attracted to one another and allowing for this kind of coiled shape to happen, okay? I could zoom in more and I'd show you right here, right here, right here. These are hydrogen bonds, or, hydrogen bonds over here, okay? But just for now, it suffices to understand that it is hydrogen bonding between these guys. Some proteins can also take a tertiary structure and a quaternary structure. Quaternary structure is a little bit more rare, but tertiary structure, for example, is where you have this mix of alpha and beta. It's a 3D structure for the protein, and it is being, it really occurs because of some hydrophobic interactions and covalent bond interactions, okay? For example, one of the most common ones is a disulfide bond. And just to demonstrate to you, do you guys know like the amino acid cysteine, okay? Cysteine usually has this sulfide group, okay, that looks like this. If I take two cysteine molecules, okay, let's say there was a cysteine molecule right here, and there was another cysteine molecule right here, okay, and it was holding this protein in this shape by a bond. This would be the bond. How is that bond looking like? Well, I have two cysteine molecules, so here we go, cysteine, cysteine, another thiol. And what ends up occurring is that these guys right here will leave and these two will connect, okay? They call that a disulfide bridge, okay? This is a covalent bond. This is a very strong bond, okay? Nice. So that's a disulfide bridge. And again, covalent bonding and hydrophobic bonding can determine the tertiary structure of a protein. And you can see, again, it's consisting of alpha and beta pleated sheets here as well. Now, a quaternary structure is interesting. It is actually not a single protein. It is one or more protein or polypeptide coming together to make one functional giant protein. And you can see here, like it's got one subunit, it's got two subunits that come together to work and create a multimeric functioning protein. A good example of this is hemoglobin, where it actually has four subunits, right? You guys know it's got the alpha 1, the beta 1, the alpha 2, and the beta 2 globin chains, globin peptide chains, right? And they function together to carry oxygen, carry CO2 and hydrogen and other stuff. Okay, so now you guys know about all of this. One more thing that you should technically know about is that amino acids, they're such interesting things in the body, they can be transaminated. What does that mean? What does that mean? Okay, that means that I can take an amino acid, okay, for example, alanine, okay, I can take an amino acid like alanine, and I can move its amino group, right, that amine group right there, and I can move its amine group elsewhere on another molecule to form another amino acid, okay? So I can take one amino acid and I can form another amino acid from it. What do I mean by this? Well, I have one alanine right here, and I can take that alanine and make another amino acid called glutamate. How is this done? This is a transamination reaction, and it can only be done in the presence of something called a keto acid, okay? Or an alpha keto acid, I should say. A keto acid, the easiest way to think about it is it's a carbon backbone skeleton, okay? It's a carbon backbone skeleton for an amino acid. It just lacks the amino group. What do I mean? Well, take an example here. Alpha-ketoglutarate, which if you guys remember, it's an intermediate in the Krebs cycle. It's a keto acid. Look at its structure. Look at its structure. Isn't it oddly similar to the structure of glutamate, the amino acid? It really is. It is the exact carbon backbone skeleton. What is it missing? The only thing it's missing is this ammonia, okay, or this amine group. So what do? We go through a transamination reaction where we take one amino acid like alanine, okay, and we transfer that amine group on the alpha-ketoglutarate, okay? And the resultant, so look, we're going to go this way. We're going to take these two, we're going to react them one another, and we're going to get glutamate and pyruvate. Okay, what is pyruvate? Again, if you guys remember, pyruvate is still part of the glycolysis chains, okay? It's the last thing we make before we go into the Krebs cycle. But why is this kind of important right now? Pyruvate is another alpha keto acid, which means what? Which means I can actually reverse this reaction. I can take the glutamate, right, and then take the pyruvate and make alanine. All right? So this is a transamination reaction. It's actually very, very commonly occurring in the body. All right. And again, you can see the structure of alanine strikingly similar to the structure of pyruvate, but pyruvate is only missing the amine group. Okay. In fact, they both have this exact same carbon backbone skeleton. Okay. And you'll see the reason why I highlighted these is because that's the R group. You guys remember how we said amino acids have an R group, the variable group? Well, for alanine, it's this guy right here. And for glutamate, it's this guy right here. And you can see that the respective keto acids will have the same thing. That's why they can be transaminated with one another. Alrighty. Now, this is not the only transamination reaction that can occur in the body. There are other amino acids that can be transaminated. Usually, when there is a transamination reaction, there is an enzyme called a transaminase. For example, the enzyme in this scenario would be alanine aminotransferase. And funnily enough, as a cofactor for that enzyme to work, they use pyridoxal phosphate, which is vitamin B6. There are other reactions that can occur. For example, there's like decarboxylations, deaminations, deamidations. We're not going to talk about that. I'll briefly touch on a deamination in, believe me, the simplest form possible. And for example, that's when you take an amino acid. Okay, I'm not going to go into detail, but you'll take an amino acid. Okay, here's the ammonia, here's the carboxyl group, the R group. Okay, and here's the alpha carbon. And you strip this amino acid of its ammonia, forming just this free ammonium, okay, which is very toxic, and then a keto acid, right? Because remember, all a keto acid is just a carbon skeleton of an amino acid without the ammonia. So you can form a keto acid. And you can use this keto acid for, many other things. You can make glucose from it. You can make ketone bodies from it, or you can oxidize it. Doesn't matter. Either way, once you make this ammonia, you're going to take it into the liver, okay? And then in the liver, this guy will go through something called a urea cycle, where it's basically just a way of getting rid of this ammonia because it's super toxic in our bodies. So we want to get rid of this ammonia, and we create this molecule instead called urea. And the urea will then go into the kidneys and then get excreted out as urine, okay? So it'll get urinated out. But all a deamination reaction is we're taking the amino group off of an amino acid and we're putting it into a urea cycle, which occurs in the liver. Awesome. The next thing I want you to know is all the different types of amino acids that we actually have, okay? This one will take a little while, but don't fret. So, you can classify amino acids in many different ways. You can classify them by their function, or you can classify them whether or not they have a charge, okay? Are they net positive? Are they net negative? You can also classify them as whether or not they like to interact with water. So polar amino acids, they like to interact with water. Non-polar amino acids, they don't like to interact with water. Now, do understand there is some controversy that can occur with some of these. So for example, cysteine. A lot of people can say that cysteine is a non-polar amino acid, but other people can say that cysteine is a polar amino acid. And that's just because of the chemistry of it, okay? So for example, some people say that this sulfide group allows it to be polar, and others say that, well, it's relatively polar, but it's more so non-polar. So again, there's some controversy that goes on there. Now, let's go through the list of all these amino acids quickly, and then we'll talk about which ones are non-polar, charged, and polar. So we have all these non-polar ones, right? So we got glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline, okay? Why do I have these little letters beside them? Okay, well, these little letters are just their one-letter code. So glycine is G, alanine is A, valine is V, leucine is L, isoleucine is I, methionine is M. Some of them are weird, though. So, for example, phenylalanine, it's not going to be P. What is phenylalanine? Well, here's how I like to remember it. Phenylalanine, this makes a F sound, so it has to be F, okay? That's how you know phenylalanine is F. Tryptophan, to be honest, I just kind of remember it as a W, but if you want, you can kind of remember the shape. I think of it as a W shape like that, but it's a W. Proline, pretty easy. It's just a P, so it's proline. Next, we're going to go to the electrically charged side chains, meaning that they are going to be either negatively charged or they're going to be positively charged. All right, so how do you know one is negatively charged, guys? Well, I'll tell you a trick. So here is a carboxyl group, right? COOH. But then here's a carboxyl group. If you guys remember, a carboxyl group is negatively charged, whereas an amino group is positively charged. So these guys are negatively charged. Okay, these guys are negative. Okay, you know another name we can give these guys? We can say they're dicarboxylic because they have one carboxyl and two carboxyl group. Okay, same thing, one carboxyl, two carboxyl group. And there's only two negatively charged or dicarboxylic or acidic amino acids. And those are going to be aspartate or glutamate. Sometimes you might see it be written as aspartic acid or glutamic acid, but it's the same thing as glutamate and aspartate. The other ones that are basic, okay, why are they basic? Because they have a positive charge here, okay? And what makes them have a positive charge? An extra amino group on their side chain, okay? If you guys haven't figured it out by now, the shaded in portions are the side chain. They're always going to be the side chain. That's what the shaded in regions are. So these guys are positively charged, and instead of being called dicarboxylic, they're actually dibasic, okay? Positively charged, dibasic, basic amino acids, all referring to the exact same thing, guys. Now, what is the one-letter code for these guys? Well, aspartate is D. Again, kind of no way to remember this. You just got to memorize this one. Glutamate, I like to think that because it ends in an E, it's E. But you'll see that this has a problem with it later. Histidine, well, it's just H, so it's got to be histidine. Lysine is a K. Now, this is how I like to remember lysine, okay? This is how I like to remember it. If you draw a line right here, it looks like a K, okay? So that's how I remember lysine as a K. And then arginine. This one, again, kind of a fun one. This sound right here, R, that's how you know it's an arginine, okay? So it makes a R sound. All right, finally, let's move on to the polar side chains. These guys, they can interact with water, and usually the fact that they can interact with water is due to the fact that they can form hydrogen bonds with the water, okay? Usually these guys will have a hydroxyl group on them, okay? So you'll see that the side chain will have a hydroxyl group, or this sulfide, and again, another hydroxyl group, okay, and so on. Now, these are the polar side chains, right? So what are the actual codes for these guys? So serine, again, S, obvious. Threonine, T, obvious. Cysteine, C, obvious. Tyrosine, because it's got a Y in it, I like to remember it stands for Y. Asparagine, it's got or asparagine, it has an N here, so I like to remember N. And the reason why I don't remember alanine like that is because alanine is just A, okay? Glutamine, it's Q. Now, this is where the problem comes in. Remember I said that glutamate ends in an E, so it has to be E, and then glutamine also ends in an E. But the reason I don't remember it like that is because when I initially learned this, I did like this. I said, well, the G kind of looks like a Q if you do this. So glutamine is a Q. That's the way I remember it. I'm sure you guys can come up with mnemonics or other things, but this is the way I remember it. Yeah. Now, we can even further classify all of these guys, okay? We can say some of them are Zwitter ions or neutral. What does that mean? That means they don't have a charge. They don't have a net charge. So remember how these guys had a net charge? Like these guys were negative because they had two carboxyls. These guys were positive because they had two amine groups. The rest of these guys, they're all Zwitter ions. So look, glycine, alanine, valine, leucine, isoleucine. I'm not going to go through all of them, but they're all of them, every single one of them that has this little purple coloring on it is a Zwitter ion. Another thing we can do is we can classify them as allopathic or not. Usually when we say allopathic, guys, we're talking about non-polar amino acid. What the hell does allopathic even mean, though? Allopathic is referring to amino acids that have side chains that are filled with carbon and hydrogen, but no functional group. Okay, what do I mean by this? Well, functional group is something like hydroxyl or an ammonia or a carboxyl, okay? And these guys don't have that. All they have is literally just a carbon bounded to a hydrogen, bounded to another carbon, bounded to another hydrogen, and so on and so forth. And you know what that would look like? That would literally look like this, okay? So if you see that pattern, that's an allopathic amino acid. Well, can you see any over here? Again, we usually said they're nonpolar. And yeah, of course you can see them. Right here, right here, right here, right here, right here, and right here. Not these guys, but right here. Okay? So you can see they don't have functional groups. Okay? They're just kind of straight or branched. And that's the key word that I wanted to bring up there. Straight or branched allopathic amino acids. What's a branched amino acid? You got 3 branched chain amino acids. You have valine, leucine, and isoleucine. And these guys are super, super important in muscle protein synthesis, especially leucine. Leucine is a big driver of muscle protein synthesis. In fact, if you guys know anything about anabolic complexes in the body, leucine is a big stimulator of muscle protein synthesis because of the fact that it can stimulate mTORC1, or the mammalian targeted rapamycin. Anyways, so that's how you know whether something is allopathic or not. Aromatic just refers to the fact that they have a ring or not, okay? This phenol ring or benzene ring, whatever you want to call it. And phenylalanine, of course, it has a ring, so it's aromatic. Tryptophan, again, aromatic. And the last one, tyrosine. So you can see there's 3 aromatics: tyrosine, tryptophan, and phenylalanine. Easy. One last thing is that technically there's one more classification where we can say there are sulfur-containing amino acids, and there's 2 of them. There's cysteine, so right here, and then there's methionine, right here. And in fact, you can actually make cysteine from methionine. If you look at the structure, it's damn near similar. It's very similar, okay? Awesome. One more thing that I do want to mention is this concept of essential amino acids and conditionally essential amino acids. So essential amino acids refers to amino acids that we cannot synthesize in the body. We have to get them from our diet. We have to eat them. Okay, if we don't, we're going to run into big, big trouble. Another thing is that we have conditionally essential amino acids. What do I mean by that? I mean that these guys are not essential, but in a scenario of like illness, okay, or malnutrition, they might become conditionally essential, okay? They might become essential. That's why it's conditional. All right, it depends on the conditions. If you're malnutritious, if you've got some sort of disease, if you've got like cachexia, they might become essential for you, okay? But usually the essential amino acids are the ones that we do not make. And they include, and I've put them in their 1-letter code here, they include isoleucine, leucine, this one is tryptophan, right? W. We got lysine, again, K. We got phenylalanine for the F. We got histidine. We got methionine. And we got threonine, guys. Threonine. Remember, there's 3 T's. There's tyrosine, tryptophan, and threonine, okay? But threonine is the one that has the T 1-letter code. And these guys are all essential amino acids. Another special thing is that technically lysine, histidine, and threonine, they cannot even be transaminated. So the rest of these guys, if you find their keto acid in the body, they can be transaminated to form methionine or, phenylalanine or the rest of these. But these 3, they can't even be transaminated, so they're even more essential. Last thing is these conditionally essentials, and they involve proline, okay, which is right over here, okay. The last one is arginine. You guys remember that? Arginine. Y, tyrosine. It's got a Y in it. Where's tyrosine? There it is, tyrosine. And then C for cysteine, and then Q for that glutamine, okay? So these guys can become conditionally essential if, again, some sort of disease is about. Now, guys, that's going to finish it off with this video. We talked a lot about the structure and all this stuff. If you really, truly enjoyed it, if you learned something from this, if it helps you, please consider subscribing. And I'll see you in the next video when we talk about the digestion and absorption of amino acids.